Oops. Let's go back to there. I probably can't get in there. Okay, before we begin, are there any questions? Somebody confirm you can hear me?
Yeah, we can hear you.
Great, thank you. Let me share my screen. All right, let me restate. Are there any questions before we begin? Excuse me a minute. Hopefully you can see my screen. Today is March 5th, and we're supposed to be, wait a minute. We're supposed to be starting chapter 13 today. We're still on chapter 14, so we're behind schedule. Let me remind you, there's going to be a quiz tomorrow, this Wednesday. It's going to cover chapter 8 and then lab 9. I'm going to try and work on getting lab 9 graded tonight. So probably not after the lecture, but maybe after the lab. Hopefully I'll have lab 9 graded. Any questions? If not, let's move on to chapter 14. Let me state that there is no lab today. I will be logging in for the first 15 minutes to answer any questions, but after that, I will be logging out. So if you have any questions, make sure you're in the lab for the first 15 minutes. Let's move to chapter 14. I've got that up here someplace. We had talked about the occurrence of disease, talking about some different terms, describing, well, I guess the occurrence of a disease, like an epidemic and a pandemic. Any question about any of those? If not, let's move forward. In talking about disease, it's important to discuss the severity or duration of a disease. It's another useful way of defining the scope of a disease to talk about its severity or duration. We have acute diseases, and these are diseases where the symptoms develop rapidly, the disease is short-lived, and then the disease goes away. The best classic case of an acute disease is the human cold, where you get the virus, shortly thereafter, like 3 to 4 days, you get symptoms which develop rapidly. The disease is short-lived, and then it goes away. So that's an acute disease. A chronic disease is a disease that develops slowly, persists for long periods of time, and usually gets worse with time. Sometimes the body never throws off these diseases, like hepatitis B, if you have, what is that? Recurrent liver inflammation, hepatitis. Every time you get it, your liver is damaged further, and the chronic disease just gets worse and worse. There are other diseases which get worse for a period of time. The patient gets sicker and sicker, but then eventually the patient starts recovering. A case like that would be mononucleosis, which is considered a chronic disease because it develops slowly and persists for a long period of time. Any question about any of that? There are diseases in between acute and chronic. They're called subacute diseases. I'm not going to discuss them. We're not going to be tested on them. And I'm sure none of you are familiar with Or I imagine none of you are familiar with a subacute sclerosing panencephalitis. That would be a subacute disease. Any question about these? All right. The extent of host involvement does determine the, well, I guess the course of a disease. Infections can be classified according to the extent to which the host body is affected. A local infection is a pathogen that's limited to a small area of the body. For example, a boil on the skin being caused by Staphylococcus aureus is a local infection. An abscess is a local infection. That differs from a systemic or a generalized infection, an infection that can go throughout the entire body, such as the measles. When a patient, usually a child, but when a patient gets the measles infection, they get it on more than their skin. It is carried by the blood, and the virus is growing in many different cells in the body. So it's a systemic infection. A focal infection is a systemic infection that begins as a local infection, but then spreads, like MRSA that begins as a skin infection and it spreads to the blood. The blood can carry it throughout the body. Any question about any of those? Usually, if the blood carries it throughout the body, it's a systemic infection at that point, but it started as a focal infection or a local infection. That's why I call it a focal infection. A bacteremia is the presence of bacteria in the blood that differs from septicemia, which is the growth of bacteria in the blood. Usually, when clinicians sample the blood, they're only looking for bacteremia, but in a microbiology lab, they can determine if the patient has septicemia. Both are bad, by the way. Toxemia is the presence of toxins in the blood, and viremia is the presence of viruses in the blood. The state of the host resistance does determine the extent of an infection. A primary infection is an acute infection that causes the initial illness. The primary infection could give rise to a secondary infection, which is an opportunistic infection after the primary infection has weakened the host's defenses, allowing the secondary infection to occur. So the primary infection predisposes the patient to the secondary infection. And the secondary infection is often an opportunistic infection, meaning a microbe of our normal microbiota, which then goes in and causes the secondary infection. A subclinical disease is when a patient has no noticeable signs or symptoms. So they have an inapparent infection. And we have talked about that before. And we call that what's the other term for that? Asymptomatic disease, where the patient has no signs or symptoms of a disease. With COVID-19, young adults and children, actually about 50% of them will have subclinical disease or asymptomatic COVID-19 infection. They won't know that they are infected with COVID-19. And they have no signs or symptoms, so it's no big deal to them. The trouble is they can then spread COVID-19 to other people, including like their grandparents, and that would be a very serious disease if their grandparents were to get it. Some people can actually carry a polio virus, but never have any signs or symptoms of the polio disease. So there's many different diseases which can have subclinical disease carrying patients. And their danger is they can carry the disease to someone else and then spread the disease. Any question about any of that? All right. In talking about the severity or duration of disease, it's important to talk about what a latent disease is and what herd immunity is. A latent disease is a disease where the disease, which at one time was causing the disease, goes latent, sort of in hibernation, does no longer cause disease, but it can come out of latency and then later cause disease. An example of this is cold sores, which obviously is causing the disease when you have a cold sore, and then the virus goes latent, and it can come out later, causing disease again, usually more cold sores. And this disease has a trigger, and the reason why we call them cold sores is oftentimes the trigger for this is having a cold. Cold sores are actually caused by herpes virus type 1. Another example is, maybe a better example of a latent disease, is the child gets chicken pox. And then the virus goes latent, and it stays latent for perhaps decades. And then the child is no longer a child, is no longer even an adult, it's an elderly adult. And the virus comes out of latency and then causes shingles. Of people who had chickenpox, about 15% of them will develop shingles, usually when they're elderly. Chickenpox usually hits children, although it can hit adults. And it's more serious if it does hit an adult. But chickenpox is a fairly mild disease among children. And like I said, it will stay dormant for decades. And what happens is in the reason why this virus did this is when the child is now a grandparent and then they have shingles, they will then go and see their grandchildren and then spread the virus to their grandchildren. And that's the way this virus tended to spread. Obviously, it can spread from child to child when they have chickenpox as well. Anyways, that's a latent disease. Any question about that? Where the disease goes latent and there is no signs or symptoms of disease. Actually, the virus is just sitting out. It is not replicating. It is not metabolically active. It's just sitting out, waiting to come out of latency again. Herd immunity is when many individuals in a population are immune to the disease, and therefore they can protect people who are not immune because the disease can't spread. Normally, let me blow this slide up. When a new disease hits a population, there's no immunity in the population like what COVID-19 first was when it first appeared. Nobody had any immunity to it. And then when somebody got sick, they spread it to other people. And then a whole bunch of other people came down with a disease. And that was a fear that COVID-19 would essentially spread to everyone and to especially the elderly and the sick and infirm. It was a lethal disease. If, on the other hand, the majority of the population is immune, then the only people who can get the disease are people who are in close association with people who have the disease. So that this individual here and this individual will become infected, but the disease can't spread to these non-immune individuals because it has to spread to this individual or that individual before getting to there, and it can't get a hold in this individual because they're immune. And that's the whole point of getting vaccinated, is to boost the population so that the population will have herd immunity, and then the non-immunized individuals are protected. Now, in the case of measles, you need over 90% of the population to be vaccinated, or else measles will spread. And every once in a while in the US, measles begin spreading. I think it actually was this winter, but maybe it was last winter. Measles was spreading because parents were saying, oh, I don't need to get my children vaccinated. I'm an anti-vaxxer. Measles is very rare. I'm going to risk it. My child won't get it. And then they get it. A frequent case of when measles occurs, and this has happened a couple of times at Disney, is you have a high population of children altogether, including high numbers of children who are non-vaccinated, and one child comes in with the measles, and then it spreads. Like I said, you need a very high vaccination rate to keep measles from spreading. And usually in the United States, we have at least 10% of the children who are non-vaccinated to measles. With COVID-19, you need about 85% of the population to be vaccinated, or else it will spread. And we're nowhere near 85%. I think the highest we ever got was the first vaccination. And I think maybe about 70% of the US population have that. And of course, that first vaccine no longer protects against later COVID-19 variants. So COVID-19 will continue to spread in the US population. There's no question about that, because the immunity isn't high enough, and we do not have herd immunity to COVID-19. It is true that we do have a lot of natural immunity to COVID-19 from people who have caught it. The trouble is, COVID-19, when you get it naturally, the immunity isn't that good. And that's why some people have gotten COVID-19 like 4 or 5 times. And like I said, for some reason, this virus does not establish a very strong immune response to it. And so once you get it, you do have a short window where you're immune, but then the immunity weakens, and then you can get COVID-19 again. Anyways, the whole point of getting the vaccines is to get the population to have herd immunity, and then the disease can't spread. Any question about any of that? All right, let's talk about patterns of disease. Normally, a disease follows a specific sequence of events associated with an infection by a microbe, and then the subsequent development of disease. First, the disease has to come from a reservoir. Excuse me. So a reservoir must exist where the disease comes from. The pathogen must be transmitted to a susceptible host. The pathogen has to invade the host and has to multiply within the host. The pathogen has to injure the host. If there's no injury to the host, the pathogen will not have any signs or symptoms of disease. The illness then will end when one of two things happen. One, the host immune system destroys the pathogen, and then the disease ends. Or two, the pathogen kills the host, and then the disease ends. I suppose you could have a disease which never goes away, like HIV and some AIDS patients. In fact, the virus will always be in those patients, but with modern medication, you can keep the patient alive. So the patient is always potentially sick, but they usually get better, and then they can get worse and get better. And I guess that would be a third case too. So the illness ends when the host immune system destroys the pathogen, which happens with the colds and the flus, or when the disease kills the patient, or like I said, I guess you can have the disease continuing to exist in the patient. There are predisposing factors that make the body more susceptible to and therefore affect the occurrence of a disease. For example, females have a higher incidence of urinary tract infection than males, and this is because they have a shorter urethra than males. There are inherited traits, sickle cell anemia, or the sickle cell gene, that make the patient more susceptible to getting disease. Another trait, which is not exactly an inherited trait, but diabetics are more susceptible to getting almost any disease. Climate and weather are predisposing factors. Everybody knows that in the late fall, the early spring, and the wintertime, you're more likely to come down with a cold than the flu. Fatigue predisposes you to getting diseases. College students frequently don't get enough sleep, so they're fatigued. So you got to be careful because you're at more greater risk to predisposing your body to coming down with the disease. Age is a predisposing factor. Let me just briefly show what normally happens. Oh, why didn't that come up? Let me try that again. There it went. Normally, for most diseases, so this would be our age and then time on the x-axis. Most diseases have this type of a pattern where the very young are more likely to get disease, and then the very elderly more likely to get disease. And then the lowest point will be young adults, maybe teenagers, least likely to get disease. And it might be that they're more likely to be asymptomatic, but that's what most diseases follow. With COVID-19, it isn't that way. With COVID-19, the very young have an increased risk, very low risk at young age, and then it starts rising. And then at elderly, much greater risk. So COVID-19 is very different. Only infants have a risk of getting COVID-19 and then severe death after a few months old. I don't know exactly where the cutoff is because obviously there is no cutoff. It's a decline. But young children have a very low rate of COVID-19 infection, or at least with signs and symptoms. Any question about any of that? So COVID-19 is a little unusual, and that is it doesn't follow this normal pattern where both the young and the elderly are at risk for getting disease. Lifestyle can greatly affect your predisposing factor. It's well known that smokers are more likely to come down with a respiratory illness, and that's because smoking damages their mucous membrane, as well as probably weakens their immune system. Chemotherapy is a predisposing factor, mainly in that it weakens the immune system. So you're more likely to get an infection, and the infection is more likely to get established and then cause disease. Any question about any of these predisposing factors? All right, let's move on to talking about the development of disease. Once the host defenses are breached, disease development follows a certain sequence. This sequence tends to be similar for both acute and chronic diseases. I'm going to blow this slide up. First, we have the incubation period. That is from when the patient first becomes infected with the disease-causing agent till the time when the patient first has signs or symptoms. So that's the incubation period. The prodromal period is the period when the patient first has signs or symptoms, but the patient's signs and symptoms are mild. Like when you say, gosh, do I have a sore throat? And you have to think about it. And you say, yeah, I'm coming down with a sore throat. That is the prodromal period. The period of illness is when you have the most severe signs and symptoms, like when you don't have to think about, do I have a sore throat? You say, geez, I've got a burning sore throat. The period of illness. During the period of illness, the signs and symptoms are usually the most severe, and the number of microbes in the patient are also the highest. It's followed by the period of decline, where the signs and symptoms get better. Like if the patient has a fever, the period of decline begins when the fever breaks. The period of decline is followed by the period of convalescence. The period of convalescence are when the disease-causing microbes drop below a threshold number, and then the patient has no longer signs or symptoms of disease. Note in the period of convalescence, the patient still has the disease-causing organisms. They just no longer have any signs or symptoms of disease. Any question about any of that? Now, when you have the cold, I frequently have done this. When I notice I'm coming down with the first signs and symptoms of a cold, I get lots and lots of rest. When you're sleeping, your immune system is its strongest, it's at its best, and I can actually decrease the number of microbes so that I go from the prodromal period of having a cold to the period of convalescence, because I just get lots and lots of rest. The trouble is, after I no longer have any signs or symptoms of having the cold, I don't get lots and lots of rest, and frequently the cold signs and symptoms will come back on me. And then I said, oops, I've still got the cold virus. I got to get more rest. So with the cold, you can do this. Get the illness to move from the prodromal period to the convalescence period by simply getting lots and lots of rest. I've tried doing this with the flu and it does not work. The flu is not one where getting lots of rest will make the flu go away, making it asymptomatic. Any questions about any of that? Oops, didn't mean to do that. Let me stop that. Go back. Now, you do not always see all of the stages of disease. As I mentioned, I can sometimes be in the incubation period of the cold, be in the prodromal period, and then move it from the prodromal period to the period of convalescence. In some diseases, there is no prodromal period. So you're going around fine, no signs or symptoms, and then it feels like somebody's hit you over the head with a sledgehammer. Sometimes when you get the flu, there is no prodromal period. It moves from the incubation period to the period of illness. With some diseases, there is no period of decline or period of convalescence. Before the advent of modern medicine, AIDS patients frequently were in the period of illness, and then that was it. They died. There was no period of decline. There was no period of convalescence. So not all diseases have these different periods of illness, but most of them do have all of these different periods of illness. Here's a question for you. During what period of illness are you not capable of spreading disease? So what's the word I'm looking for? Non-communicable. So what period of disease are you non-communicable? Anyone have a guess?
Is it during the decline?
That's a good guess. Anyone want to have another guess? Meaning, no, that's not right. But anyone have another guess?
Period of convalescence.
Period of convalescence. That's an even better guess. However, both of you are wrong. In all of these periods, you have the disease-causing organism. So you can spread disease in every period of illness. Now, that was a trick question. I didn't expect anyone to get it right, but it is something I'm trying to emphasize, and that is you can spread disease as long as you have the disease-carrying organism. All right? I won't ask you that on the exam, but it's something you should know because a lot of people get this wrong. My mother always told me, "Oh, I'm getting better now. I won't be spreading it." And I would tell her, "Mother, you're talking to a microbiologist. I know better." Okay. So even in the period of convalescence, you can spread disease. Even in the period of incubation, you can spread the disease. Now, maybe like a few hours after you first entered the period of incubation, you maybe cannot spread disease very, very early in incubation because the disease-causing organism has just gotten established in you. It's hardly grown at all. However, for some common diseases, the most, at least of adults, the most frequent chance of spreading the disease is late in the incubation period or in the prodromal period. This is true for colds and for flus, that for adults, you're more likely to spread the disease late in the incubation period or in the prodromal period. The reason why is in an adult, in the period of illness, the immune system has ramped up and is attacking the microbes. And so you're less likely to spread the disease, at least the colds and flus, in the period of illness than you will in the incubation period and the prodromal period. I have a theory for why that's the case, and that is I'm guessing that in our human ancestry, like our cavemen, they didn't want to spread colds and flus to their children. So their immune system ramped up in the period of illness and then made them less likely to spread the disease than before the immune system responds. That is only true of adults. For some reason, among children, that is not true. So among children, they can spread disease at all of these times, especially in the period of illness. It's because their immune system isn't ramping up and attacking the microbes, at least common illnesses like colds and flus. In the old days, now this is when I was a child, they did not allow children in certain hospitals because they realized that children have a lot of illnesses and that they're more likely to spread disease than an adult. I don't think that's the case anymore because they want children to go like see their grandparents because of the social benefit. But I could be wrong. Some hospitals may not allow children to visit. I don't know. But when I was a child, that was actually something very frequent. I was not allowed to go visit my brother who was in the hospital because I was under age 13. Any question about any of that? I think I've talked about everything I wanted to here. So let's move on. Let's talk about spreading an infection. An infection has to come from a reservoir, a source of infection. Reservoirs are continual sources of disease organism. A reservoir can be living or non-living. Human reservoirs are very common for human diseases, where the disease comes from a human. And humans often spread disease, so human reservoirs are very common. For some diseases like AIDS, gonorrhea, the only reservoir is another human. Sometimes carriers may have inapparent infections or latent disease, in which case they could spread it and they don't even know that they have the disease-causing organism. The best example is COVID-19, where about 50% of young people are a reservoir. They don't know they have the disease-carrying organism, but they can spread it. There are animal reservoirs for some diseases, like rabies. That disease only comes from an animal. Humans cannot spread rabies. A Lyme disease also spreads from the bite of a tick. Zoonoses are animal diseases that may be transmitted to humans. So a zoonosis is an animal disease that may be transmitted to a human. Like rabies is mostly an animal disease. If it does infect a human, it's actually a dead end for the virus because the virus never leaves the human to spread to another animal or person. That's not true for a dog. Obviously, a dog can spread it to other animals and people. And most animals can spread it. There are non-living reservoirs like lettuce, soil, water. And these will be examples for diseases like botulism and tetanus, where they normally come from a non-living reservoir. And then lettuce frequently spreads gastroenteritis in the United States when the lettuce is contaminated with a microbe. Any question about a reservoir? Transmission of disease is our next topic. There are three principal routes by which a causative agent of disease can be transmitted from the reservoir of infection to a susceptible host. First, there can be contact transmission. There can be vehicle transmission, where a vehicle gives the agent from the reservoir to the healthy individual. And then there can be vector transmission. A vector is an animal which spreads the disease-carrying organism to the patient. Any question about that? So vehicle is an object, vector is an animal. In contact transmission, there are three types. There's direct person-to-person transmission, indirect, and then droplet transmission. We'll talk about that in a minute. Direct person-to-person requires close association between the infected individual and the susceptible host. For example, a handshake is direct contact transmission. Kissing, another example of direct contact transmission. Indirect contact transmission is where the disease spreads from the reservoir to the susceptible host via a non-living object. We have that object, a term for that, and we call it a fomite. For example, when somebody's got the cold and then they touch a dollar bill and then they turn in the dollar bill as cash, somebody else touches that dollar bill and then they pick up the cold virus and then later get the cold. So that's indirect contact transmission. Money actually is a problem, but contaminated tissue, contaminated baseball, like if somebody is playing baseball and they have a cold or the flu, They will touch the baseball and then put it on the baseball, and the baseball will be picked up by somebody else, another teammate or foreign player, and then they'll get the disease-causing agent. Droplet transmission is transmission via airborne droplets, and these airborne droplets travel less than 1 meter. Note that is different from airborne transmission. Droplet transmission shown here, where that's about 1 meter, and there is a higher concentration of droplets within that meter, but that's droplet transmission. Direct, the high five, handshake, hand-to-hand touching. Indirect, the baseball having the disease-carrying organism, and then the catcher picks it up with that hand, getting the disease-carrying organism on that hand, and then usually the hand will touch a mucous membrane in the face and then infect the catcher. Any question about any of that? I'm not gonna talk much about droplet transmission or test you on that, because I find that 1 meter distinction a little bit of an arbitrary line. You can get infected by breathing in the mist beyond 1 meter. It's just you're more likely to if you pick it up from 1 meter, and that's what droplet transmission is considered, contact transmission. Beyond 1 meter, it's considered airborne transmission, and that is not contact, direct contact. It's considered vehicle transmission. And let's talk just a little bit about that, and I'll come back to talking about vehicle transmission. So airborne droplets that travel more than 1 meter from the reservoir to a new host is considered airborne transmission, and you can get infected from that. And that's why I'm not going to quiz you, because droplet transmission and airborne transmission, they're very similar. It's both the same sneeze that you're getting it from. It's just the length of your distance away from the patient or the sick individual. The other cases of vehicle transmission is transmission by an inanimate reservoir, usually an object that we call a fomite, and that could be food, water, or body fluids. There are 3 different types of vehicle transmission. We've already talked about airborne transmission, but there's waterborne transmission, where the microbe can get in the water, contaminate the water, such as contamination by sewage. This frequently happens in third world countries. It doesn't happen really in first world countries. But waterborne transmission is a problem in the world, just not in the first world countries. Foodborne transmission, it usually happens when food is undercooked or poorly stored, or when it is unsanitarily handled by a food worker. Foodborne transmission. And I guess if somebody were to touch the cup and then somebody were to drink out of it, I don't know, I guess that would be waterborne transmission if they're water. And if they're eating food out of the cup, it would be foodborne transmission. The third, airborne transmission, we've already talked about. Any questions about vehicle transmission? Vector transmission is when an animal carries the disease-carrying organism to the susceptible host. The most important group of vectors are biting arthropods, especially fleas, ticks, and mosquitoes, which can transmit a large number of diseases to humans. Vectors can transmit disease by 2 ways: mechanical transmission or biological transmission. Mechanical transmission is where the animal passively transport the disease-carrying organism, like when this fly lands on this object here, picking up the disease-carrying organism on the feet of the fly. Passively, the fly then transmits the disease-carrying organism to the burger, and then the burger is eaten by the patient, and that is this patient has gotten disease-carrying organism by mechanical transmission of a vector transmission, passively by the feet of the fly. Biological transmission is a little similar but different, meaning it still involves a vector, but the pathogen is reproducing inside the vector. An example of this is malaria reproduces inside the mosquito, and then the mosquito bites the human, and then malaria is transmitted to the human. So biological transmission is an example of a mosquito spreading malaria to the human. Any question about vector transmission? All right, if not, let's move on to nosocomial infection. Since most of you are going to go into the healthcare industry, sorry, this is one of the most important topics I can teach you in microbiology. What nosocomial infections are and how to prevent them. Nosocomial infections are also called health care acquired infections. Or hospital acquired infections. These are infections acquired as a result of a hospital stay or a patient staying in a hospital-like environment, like a nursing home, and the disease-carrying organism was not present or incubating in the patient before the time of admission. About 5 to 15% of all hospital patients acquire a nosocomial infection in the United States. That's 2 million people per year, and about 20,000 deaths per year in the United States alone. This used to be the 8th leading cause of death in the US. It might be the 9th now because COVID-19 has become a cause of death. So it is a serious disease, nosocomial infections. About 1 out of 10 hospital patients gets a nosocomial infection. This is from the CDC. About 50% of nosocomial infections are preventable. And then there are major economic costs to the United States. You don't need to know that, but I'm just going to briefly mention that. Nosocomial infections result from the interaction of 3 factors in the hospital. First, microorganisms are present in the hospital or hospital-like environment. Hospitals are major reservoirs of sick patients, and these sick patients frequently have nosocomial infective agent or some other agent which can spread and cause disease. So microorganisms are present. Some of the nosocomial infections are actually the normal microbiota that become opportunistic pathogens. And they can be spread by the staff as well as patients or visitors in the hospital. And then some nosocomial pathogens are antibiotic resistant, and that causes a problem. So when the patient gets the nosocomial infection, the doctor prescribes the normal antibiotic, and lo and behold, they're resistant to that antibiotic. The doctor then prescribes a second antibiotic and orders a test to determine which antibiotic to use. But by this time, it might be too late. The patient might be dead from the nosocomial infection. Okay? Any question about any of that? The second reason why nosocomial infections happen in the hospital-like setting is the patient is a compromised host, one whose resistance to infection is impaired, either by disease, by therapy, or burns. The patient is in the hospital for a reason, and they're usually a compromised host if they are in the hospital. They may have broken skin, broken mucous membranes, compromising the host, burns, compromising both injections, ventilators, urinary catheters, the administration of anesthesia, all can help compromise the host. And then the patient may have a suppressed immune system. First, they may be sick, so the immune system isn't going to be strong to begin with. They may be taking drugs, which will suppress their immune system. They may be having radiation therapy, which will be suppressing their immune system. Or they may have a burn, which is suppressing the immune system. So they'll be more susceptible to getting the disease, the nosocomial disease. And then third, in the hospital or hospital-like setting, there's a chain of transmission. The principal routes of transmission are direct contact transmission, like patient to patient. In special care units, hospital staff have to make sure that doesn't happen. Hospital staff can also spread disease. So after they touch a patient, even if they're wearing gloves, they should remove the gloves and wash their hands before moving on to the next patient. There can be indirect contact transmission. This happens via fomite, like our urinary and IV catheters, contaminated surgical dressing. If anyone ever watched the old medical movies, you would note that the doctor in the 1950s and 60s proudly wore the stethoscope on his chest. So he'd have a lab coat on and have a stethoscope. He'd take the stethoscope, listen to the patient's heart, and then what would he do? He would drop it and it would thump against his chest. And then what would he do? He'd go to the next patient, take the stethoscope and listen to the patient's heart by touching the skin. And they were actually spreading nosocomial infective agents with that stethoscope. They now know that that's a concern, and that if they do that, they have to sanitize the stethoscope before moving on to the next patient. Indirect contact transmission. Vehicle transmission, such as airborne transmission, concern that the ventilation system may spread the disease. For cases like a child's hospital, measles is a concern that it might spread by the ventilation system. They initially worried when Ebola patient, when somebody who was working in Africa with Ebola patients, and then they had inappropriate handling of the Ebola patients, and so they became sick, and they came back to the United States to receive care. There was concern in the hospital that the ventilation system may spread Ebola. And so what they did was they put the patient in a special unit and then cut the circulation from that room so it wasn't spreading throughout the rest of the hospital. I'm not sure that Ebola actually spreads by the ventilation system, but I'm just saying that was a concern. Any questions about the chain of transmission? There are certain procedures that are generally implemented to prevent the spread of nosocomial infection. So when you're working in the healthcare industry, you have to control nosocomial infections. First, you've learned aseptic technique in microbiology. Now, when you're working with the patients, you always follow using aseptic technique. You need to properly handle contaminated materials. You have to frequently and thoroughly wash your hands. Like after each patient, if you aren't wearing gloves and you don't dispose of the gloves, you should wash your hands before moving on to the next patient. There's education of the staff, the patients, and the visitors, and hospitals have gotten very good about that. In the 1960s, like I said, they didn't even educate the doctors, let alone the staff, and certainly not the patients and the visitors. But in the last 10 years, they're educating the patients and the visitors. And you'll see in the hospitals, they have signs outside the hospital room or outside the elevator saying, sanitize your hands, prevent the spread of disease. And they'll have a sanitizer either in the elevator or outside the elevator and outside each patient's room. And that's to prevent the spread of disease, especially nosocomial infectious agents, but other disease agents as well. There are many other ways to control nosocomial infection agents. See the online chapter notes for further discussion of that. Any question about nosocomial infections? As I stated, they are a major concern in the hospital. And if you work in the hospital, you need to work on reducing the spread of a nosocomial infective agent. As I stated, 50% of the patients who get a nosocomial infective agent, that could have been prevented if people had been educated and followed through on preventing the nosocomial infective agent. Any question about any of that? All right, let's move on to epidemiology. Epidemiology is the study of where and when disease occurs and how diseases is transmitted in a population. The CDC collects and analyzes U.S. epidemiological information. Usually what happens is the doctors report a reportable disease to the county health authorities. The county health offices then report it to the state offices, and then the state reports the state offices to the CDC. Now, in the state of Washington, I don't know where the county offices are for the health department. I just don't know where it is. In Oregon, which I used to live in Oregon, it's in Salem. So the county, which I lived in Washington County, so the doctors would turn it into Hillsborough, the county local health offices. I don't remember what you call them, but, and then they would send it into Salem, and then Salem would then send it into the CDC. In Washington, I don't know where the state offices are. In Clark County, I'm sure they send it into Vancouver, and then they send it into the state offices, and then they send it into the CDC. The CDC office is actually in Atlanta, Georgia, and then they collect all of that information, analyze it, and then publish it weekly in the Morbidity and Mortality Weekly Report, which is usually called MMWR. And you can find that at the Centers for Disease Control at www.cdc.gov. Notifiable diseases are those diseases that physicians are required by law to report. And each state has a different list, but generally most states have the same organisms which are notifiable. Any questions about any of that? All right. Morbidity is the incidence of a specific notifiable disease. The morbidity rate, the number of people affected in relation to the total population in a given time period. The mortality is the deaths from a notifiable disease. The mortality rate, the number of deaths from a disease in relation to the population in a given time period. Any question about any of that? Any questions about chapter 14? If not, let's get started on chapter 13. I don't know if I saved that or not, so let me save that. I mean, I don't know if I changed. I got to pull out the, let me leave that there and I'll keep an eye on that. Chapter 13 is viruses, viroids, and prions. There he is, just a little slow here. This slide is showing you the major goals and rough outline. Know the terms always. If you want to know all the terms, you should get the terms from the objectives, not from here. These are just the major terms. 2, understand the characteristic of virus structure, virus classification. And 3, understand viral multiplication. Viruses have different ways of multiplying, and we usually call them a life cycle. We'll talk about different viral life cycles. And then 4, understand about prions and viroids. Any question about what we're going to cover? The only goal a virus has is to replicate. How do viruses do that? They generally invade a host cell, hijack the host cell, and then cause the host cell to replicate the virus. So let's watch a little video on that. Turn that down briefly. Phage is a short for bacteriophage. We'll talk about what they are. They're bacterial viruses.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
Any questions about that movie? All right, let's talk about some general characteristic of viruses. Viruses are inert when they are not in their host cell. That means they do not display little or no metabolic activity. They're not replicating. They're essentially not doing anything when they're not in their host cell. Viruses require a living host cell to multiply. So we can say that viruses are obligate intracellular parasites. They must infect a host cell. So they are an intracellular parasite, or else they can't reproduce. Viruses multiply within a living cell using the host cell's enzymes, nucleic acid, amino acids, ATP, and etc. Viruses have no ATP generating mechanism. They rely on their host cell to provide the ATP that they need. Viruses contain DNA or RNA as their central molecule, what we can call their genome, but viruses never contain both. They either have DNA or RNA as their genome. And then viruses have a protein coat surrounding the nucleic acid as their central molecule. So here we have a virus, and it has a central molecule, which will be a genome, and that will either be DNA or RNA, never both. And that will be surrounded by a protein coat, which is shown here with this cut out so we can look inside the virus. So viruses always have those. Some viruses also are enclosed by an outer lipid envelope. That would be out here. surrounding the protein code. Not all viruses have an envelope, but some do. Any question about any of that? The host range of a virus is the range of host cells a virus can infect. This is determined by specific host attachment sites, usually called receptors, that are on cell walls, flagella, or other parts of the cell, as well as cellular factors necessary for viral multiplication. Usually there are molecules of the virus that are called ligands or adhesions that bind to a molecule on the host cell that are called receptors. And that adhesion receptor binding has to occur before the virus can attach to the host cell and then infect the host cell. Most viruses only infect the cells of one or a few species. For example, AIDS, HIV virus, only infects certain cells like T cells and macrophages and brain cells of humans, and then of one of the chimpanzees. There are 2 chimp species and only one of them can be infected by HIV. And then that's it. All other cells, like your skin cells, your muscle cells, your bone cells, cannot get infected by HIV. And then obviously the cells of your cat or your dog, they will not get HIV because it's the wrong species. It only infects certain cells of 2 species. That isn't the case for all viruses, but many viruses only infect some cells of one or a few species. And these are called the host species. An example of a virus that infects more than one species is something like bird flu, which can infect many different birds, can infect many different mammals, including humans. I don't know if it can infect other animals, but that's many different species of birds and animals that bird flu can infect. Somebody has their microphone on. If you could turn that off, please. Bacterial viruses are called bacteriophages, and these are viruses that only infect species of bacteria. A shorthand name, which I don't use, is phages or phages. I will always use the term bacteriophage or bacteriovirus. Any question about any of that? Viruses, this is looking at the relative size of viruses. Let me see if I can blow this up. So right there is a red blood cell. We're only seeing a part of it. There's an E. coli, which is slightly larger than your average bacteria. And there is the largest virus in diameter, megavirus, which is smaller than E. coli. And then the longest virus is Ebola virus, which is not the largest virus by diameter, but it is lengthwise. It's skinny, and lengthwise it's the longest virus. I think there's a new virus out, which is actually bigger than megavirus, and we'll talk maybe about it at the very end of the slide, this lesson. The smallest virus you've probably never heard of. Let me see if I can blow that up and even read it. MS2. Bacteriophage. You've probably never heard of that. However, one of the smallest viruses that you have heard of is poliovirus, and it's just a little larger than MS2. Any question about any of that? Come on, let's drop down. Let's end the show. The important point is that viruses are about roughly 10 times smaller than a prokaryote cell. Their size range from 20 nanometers in diameter to 440 nanometers in diameter. And that's looking at megavirus, the largest virus that, at least until recently, was about 440 nanometers in diameter. Viruses can be longer than 400 nanometers. Remember, that's the diameter. And Ebola reaches almost 100 nanometers. So between 20 to 100 nanometers in length. However, Ebola is not the largest virus, at least in diameter. What's the point here? Oh, viruses. The largest virus is about the smallest size of the smallest prokaryote cell. The smallest prokaryote cell is around 400 nanometers. And then the average prokaryote cell is between 1 to 2 micrometers. That would be 1,000 nanometers for your average bacteria cell. Talk about virus structure. A virion is a single mature, complete infectious virus particle. So usually when we're talking about a virus and infects a cell, we say that it makes new virions. Viruses have a nucleic acid, that is their genome, and that genome is either DNA or RNA. It is never both. It is one or the other. What's unusual about their genome is it can be double-stranded or single-stranded. Double-stranded DNA is what our cells are normally, our genome, but we can have single-strand DNA, like when the DNA is replicating or when it's being transcribed. Double-stranded RNA is unheard of in eukaryote cells. Double-stranded RNA does not happen in eukaryote cells unless the cell is infected by a double-stranded RNA virus. That's the only way a eukaryote cell has, or even for that matter, a prokaryote cell, does not normally have double-stranded RNA. Single-stranded RNA is another genome for the virus. And if we're talking about single-stranded RNA, we have to differentiate between the positive single strand and the negative single strand RNA. The positive single strand RNA is a strand of RNA that encodes AUG, and these codons can be read by the ribosome to translate the viral positive single-strand RNA into a protein. The negative single-strand RNA cannot be read by the ribosomes. It does not have AUG. It would have the corollary of AUG, which would be UAC, I believe. The ribosome cannot read negative single-strand RNA genomes. Any question about any of that? Viruses' genomes can be linear or circular. If you remember, eukaryotes have linear DNA as their genome, and then bacteria have circular DNA for their genome. So viruses can be either linear or circular, depending on the species of virus. Some viruses, like influenza virus, have their genome in several segments. Most viruses have their genome just in one segment. And bacteria generally have their genome in only one segment, so the bacteria only have one chromosome. But as you know, humans have several segments, and we call these segments a chromosome. We may or may not call the viral genome a chromosome. Generally, I don't use that term for viruses. I just use the term viral genome. But some people do use the term viral chromosome, which is correct, I guess, if the virus genome is DNA. But if the virus genome is RNA, that's not a correct term to use. And it's not really a chromosome because there may not be proteins associated with the DNA of the virus genome. Any question about any of that? All right. The capsid is the protein coat that surrounds the viral genome. So here we have a protein coat surrounding the viral genome, and that's called the capsid. The virus is composed of capsomeres. Let me go back up and blow this up. So here's the capsid. Each one of these proteins is called the capsomere. On some viruses, like this one here, you just repeat the capsomere to get the capsid. So this capsid is composed of many different numbers, but it's composed of only one type of capsomere. That isn't true for all viruses. There can be different types of capsomeres on the virus. Let me show you one. Move down. Move down. Oh, here we go. This virus here, this one here, would have at least one capsomere for the head, probably two capsomeres for the sheath, one for the capsomere for the, I can't read that, tail fibers, one capsomere for the pin, one capsomere for the base plate. So several capsomeres at least on this virus. Any question about any of that? Oops, let me end the show here. Go back to where we were. Oh, here we are. If the virus lacks an envelope, it is called a naked virus. So a virus without an envelope around it is a naked virus. An envelope virus obviously has an envelope around it, and that is a lipid layer. It also has proteins and carbohydrates in the envelope. Many envelope viruses have glycoprotein spikes on it, which, let me see if I can blow this up. So that reddish purple is a spike, usually glycoprotein spikes. These spikes usually are the antigen to the virus. An antigen is a molecule on the virus or some other microbe that our host immune system responds to. And usually the host immune system is responding to an outermost molecule, and the outermost molecule of a spiked enveloped virus would be the spikes. And so these are the antigens. The outermost molecules of the virus, not only are they the antigens that the host immune system will tend to respond to, they are also the molecules of the virus that bind, or at least tend to bind, to the host cell receptors. So the outermost molecules are both the antigen and the attachment points for the virus. Any question about any of that? Now, obviously, the outermost points of this envelope virus are the spikes. What's the outermost molecule, which is the attachment point for this virus here? Anyone? What's the outermost molecule of this virus? It's pretty obvious to me.
Capsid.
Yeah, the capsid. When we're talking about viruses, they have a general morphology, that's the shape. Viruses are classified on the basis of their capsid structure. There are helical viruses. Let me blow this up. So right there, the helix, the virus is in the helix, the capsid. There are polyhedral viruses, and these are viruses that have many sides, like a complex dice. And there's actually a picture of a polyhedral virus. And then the third morphology is a complex virus. A complex virus can take on essentially any shape. Like this is a complex virus, this is a complex virus, and this is a complex virus. If you're not familiar with that, that looks like the lunar lander of the Apollo space program. And this is bacteriophage T4 taking on that shape there, which is a complex virus. This is a complex virus. It's also an envelope virus. And there we're seeing the envelope around it. And that would be the envelope here. And it has spikes, which are there. You can kind of see the spikes. They're the whitish things showing up there. Any question about any of that? We're going to talk a little bit about T4 bacteriophage. This is a virus that infects bacteria. Why we call it a bacteriophage? Because it infects bacteria cells. T4 is a bacteriophage that infects E. coli. And probably because of that, T4 is the virus that has been studied more than any other virus, including human viruses. So T4 is the virus we understand the best because it's been studied more than any other virus. T4 belongs to the T-even family of viruses, like T2, T4, T6, and they all have this shape. Any question about any of that? If not, I'm going to stop here, and we'll continue with this lesson in the future. We're behind schedule, and so I have a feeling I'm going to change the schedule. And I'm probably going to move the review to the lab so that we can have more time to finish chapter 13 and chapter 5. We'll see how we end up on Thursday. If we do finish chapter 13, I doubt we will. I won't change the schedule, but if I don't, I will change the schedule and I'll move the review over to the lab. Any question about any of that? All right, any questions about anything? If not, I will be at the lab at the normal time for the first 15 minutes, but you do not need to show up unless you have questions.
Thank
I
you.
actually do have a question really quick
Uh-huh.
before we go. The infectious disease project, when is that going to be graded? Do you know?
I have no idea because I'm behind on the grading. I'm working on trying to get lab 9 graded so you can study that for the quiz tomorrow. So that's my priority.
Okay, thank you.
I have a quick question.
Uh-huh.
For the unknowns project,
Mhm.
I had put my what I thought my unknown was. If I had gotten it wrong, would you have told me and I just go ahead and do my project now?
I'm not going to tell you until after I grade your unknown report.
Gotcha. Okay, thank you.
All right.
Have a good night, Dr. Volpe.
All right, you too.
Thank you.
Bye.